Good morning and a warm welcome to the participants who are assembled here for the two-day international webinar on recent trends in bioscience and biotechnology. Respected Principal Madam Dr. Varalakshmi Anandakumar, Principal in Charge, Queen Mary's College, Dr. Kamalakannan, CEO, Arune Research Foundation, Tiruvannamalai, my dear colleagues, Botany Department, Queen Mary's College, and all the faculty members from various other colleges, research scholars, and my dear students. I extend a warm welcome to the resource person, Dr. Muhammad Abdul Latif Abubakar, Senior Lecturer of Applied Sciences and Technology, University Tun Hussein on Malaysia. We are in the crossroads between real teaching and virtual teaching. Digitalization is inevitable in education as in all other fields. It would have taken another few years to introduce the digital system into the Indian system of education, but the COVID-19 pandemic has forced this change upon us at a faster rate. Today we are facing a lockdown and the campuses are not open for students and faculty. The only other option is for online teaching. But education has to continue, the knowledge has to be shared so that the students are not affected by this unusual circumstance. Online teaching and webinars are advantageous in one way, where the students have the opportunity to learn in their own convenient places, convenient time, and the knowledge from abroad could be shared easily. Even after normalcy has returned, after COVID-19 scenario is brought back to normalcy, international webinars could be conducted so that we can share the knowledge from the resource persons who are abroad and cannot travel long distances to take part in the seminar. In this way, the resource scholars will be benefited by such arrangement. So even when a normal education system is returned, we can conduct international webinars so that the resource person will share his eminent knowledge and we can update our day-to-day improvements in the field of science, especially biosciences, where the advances are improving every day. I welcome all the participants, the resource person and the budding scientists for this two-day international webinar so that everyone will be benefited. Welcome sir, welcome resource person, welcome to all.
Session. I hope this can be a two-way session for all of us in which you can also help and give opinions and inputs to me. So if it's okay for me to start my presentation, Doctor?
Yes, sir. 1 second, I give you the host and you can take the shares.
Sure.
Yeah, now you are the host, sir. You can give your slides.
Sure. Okay. Okay, so can you see my slides?
Yes, sir. Yes, sir.
Okay. So this is something that is very interesting to have for all of us to see and to discuss because I'm from a wildlife background, and I think it resonates well with what I'm trying to share today because I believe majority or the major parts of bioscience and biotechnology are concentrated in the field of botany, especially, and it's very less and very the expertise, the resource and fundings are very scarce in terms of wildlife research and conservation. But I think it's applied well. The framework generally applied to almost all organisms, and of course, I hope we can learn together in this thing. So I would like to start by sharing with you. How do I? Okay, so this is Malaysia's biodiversity. I would like to give an overview of what Malaysia have. So we are divided into 13 states and also 3 federal territories, which is Labuan, federal territories, and Putrajaya. And we have currently 29 million people in the country, of course, much less than India. And with this, it comes with 40 ethnic groups, right? So Malaysia are known as one of the multi-race and multi-ethnic with a diverse group of religious and also culture available in the country. So of course, we have Malays, we have Indian also in the country, we have Chinese, we have native people of Borneo, such as Kadazan, Iban, Malanao, Dayak, Dusun, and many other native tribes. And we also have aborigines in the country, divided into three main groups, which is Negrito, Proto-Malay, Senoi, and we have more than 10 subtribes of these aborigines in Malaysia. So why I'm explaining this, because I want to give you an overview of what we have and how the impact of bioscience and biotechnological approach will help the environment, as not only in Malaysia, but also in the region as a whole. So after independence in 1957, Malaysia, which is practically the economy is commodity based, the economy is agricultural based, has diversified its economy to a robust industrial manufacturing sectors, service sectors, and so on, in which with this, it comes with a price. So in terms of biodiversity, we are one of the 12 mega diverse countries in the world. And a conservative estimate, we have 15,000 species of plants with a high degree of endemism across herbaceous plants. We have more than 600 species of corals, which constitute almost 77% of the world's coral species. We have 4 out of 7 species of the world's turtles: green turtle, the hawksbill, leatherback, olive ridley. In terms of mammals, we have 300 species. Mainly, as you can see, the Malayan tape, one of the center of conservation conflict in Malaysia right now. We have less than 1,000 individuals in the country. We have a high conflict, especially in road fields and also hunting. In terms of amphibians, for example, that is our pride of the country. That is Megophris nasuta, which is the Malayan horned frog. So we have a lot of biodiversity in the country. But with the in the name of development, in the name of economical diversification, in the name of prosperity, in the name of development, in the name of development of infrastructure, and so on, we are destroying our nature. And this is not the case only in Asia, but this is the case everywhere, especially in the Asian region, where we are developing countries. We are building our countries to a higher level, especially for the society. And I think most of the country which is located in the equator or near the equator are facing the same issues. We are in the conservation and also development dilemma, in which we are trying to develop the countries, but at the same time, we are destroying our nature. We are promoting habitat fragmentation, habitat destruction, logging, illegal trades, pollutions, and many others. So in essence, I'm not trying to say that the destructions is a total, or I would say, is the only thing that we can see in the nature around us. But I think it resonates well with all the Asian countries that this is what we are facing right now. So if people were to ask, someone were to ask, what is the importance of conservation of all these things, especially with the current. Okay, so if we, I mean, of course, coming from all the different background of botany, I would say it's not a very weird thing to listen to today, but talking to stakeholders, authorities, policymakers, they always try to understand what is the importance of wildlife. All right, so I just divided this into 4 main categories, right? In terms of biodiversity conservation itself, this is not an option. Conservation is not an option. It's a compulsory and also mandatory actions needed by the humankind, because this is our natural and also regional heritage that we must conserve, which have evolved through millions and millions, hundreds of billions of years to what we have today. So we can look at many aspects, ethical, moral perspective, and so on. We still have to conserve all this wildlife that we have around us. In terms of ecological importance, of course, we know that animals by now is an important seed disperser. They are a pollinator. The interactions, the herbivory interactions in the natural ecosystems are important for the stability and also resilience of the ecosystem, and many others. In terms of economical outputs, especially right now with the emerging diseases and issues with public health, the medicinal value of the plants have to be highlighted in the current situation, in the current era. And we cannot put, for example, the economical output in terms of tourism from the ecosystem as one of the benefit for the humankind also. And lastly, if you're talking about what is the human needs or the human benefit from this conservation, is we can look back up to the three main categories that I divided before this. And lastly, I would say that I think almost all of us will agree to this, that the environment and the nature that we have around us today is not ours alone, but it also belongs to the future generations. Last year, Malaysia actually saw, I think in my lifetime, I actually saw the local extinction of our rhinos, right? And it is a very sad thing in the episode of Malaysia's conservation due to many issues, all right? Due to many issues, not just because habitat destruction, but also to the biology of the animal itself. But we don't want to see more extinctions in the future, because at least in my lifetime, in which it just showed that I failed to do something to conserve all these animals. All right, so this is what I'm trying to share today with you. I'm just giving you an overview and introductions of wildlife just now and biodiversity. So molecular approach in understanding wildlife. Of course, talking about molecular approach, why I think this is, I'm not really, I don't really know that the majority of the audience will be from botanical background, but I would just like to extend this, that in terms of wildlife research, the opportunity, the funding, the expertise, the equipment, the knowledge and everything in terms of molecular approach in wildlife research is very limited, especially in Asian countries. We have to accept this fact. If we are from, from a biotechnological or bioscience background, we might see this as something that is not real, but we have to engage with the stakeholders. We have to engage with our neighboring countries. And this is what I felt after years and years of research, that I think the technology that existed in terms of molecular approach in conservation and wildlife is not fully utilized to help us understand the wildlife that we have today, right? I will talk more about this later on. But let's look at how we can use molecular approach to understand wildlife. So the first of all is that the greater level of comprehensive and dynamic knowledge is required for us to understand the interactions, understand the factors, understand the relationships that we have with all these subsets and components of biodiversity, either in ecological or biological or any particular levels that we want to understand. But we need to understand them. The question is right now, especially in Asian countries, I see this trend in Asian countries, those that actually trying to stand alone in terms of conservation, those that are actually closing the door on the, not closing the door, I mean, put restrictions on the entry, and also, I would say, the issue of biopiracy. I mean, posting out all the samples of the foreign countries, the invasions of foreign scientists without legal permits and requirements from the authorities. So these are something that is becoming more and more concerning in the countries itself, in which without the understanding, using molecular tools, we are actually behind in so many levels in understanding this, right? I will talk about more about this when we see the trends and expansion of this technology in wildlife research. The second one is that we have to understand that understanding the genetics diversity or the genetics elements of any wildlife, either animals or plants, that is the core business in understanding the interactions, understanding the wildlife and the organism itself, in which, if I still remember correctly, Whittaker in 1972, he actually said that genetic diversity is the fundamental currency. That is the fundamental currency in any diversity that we can have, either species or ecosystem diversity. Without species genetic diversity, species diversity, you won't have ecosystem diversity. So understanding this genetic diversity is very important. But this is the level that is mostly neglected in almost all countries, in all developing countries, because the stakeholders, because the authorities, they are happy that if that organism is still around, without even thinking about the genetic conservation genetics of the particular group of animals or plants, because that's the level of understanding and also the enforcement that we have. This is happening throughout the Asian countries, all right? So I hope I can learn more about India's perspective about this later on. And of course, using molecular approach, so we are trying to expand into molecular ecology. Molecular ecology is not a new tool. This is something that have been discussed and talked about for the last 20 years, molecular ecology. But sadly, this particular approach are not really well employed and utilized in Asian countries. I'm not saying just Asian countries, any developing countries, because like I said earlier, the lack of funding, lack of expertise, lack of, I would say, collaborations with those that are actually high skilled in this particular field are not really promoting us to stand alone in the country. So, for example, in terms of molecular, it can be something that is very simple. Tigers, for example, we want to understand the feeding preferences or the prey selections in wild tigers. Using ecological approach, you actually have to observe them, prey or feed or forage on this particular prey. Using camera trapping, or maybe if you're lucky enough, direct observations. But using molecular tools, you can have a DNA barcoding technique from the feces that you collect from the tiger itself, and you can actually pinpoint the species that are, that are being fed by that are fed by the tiger itself. And you can actually, using higher tools, maybe next generation sequencing or qPCR, you can actually quantify, right? Quantify and have the general outline of the feeding preferences in terms of the numbers itself. So this is something that is old, but is very restricted in Asian regions. Phylogenetic systematics, of course, the first thing that you have to understand when you're trying to study any organism, plant, animal, any group of organism in terms of biodiversity, you have to understand the taxonomy. So the problem with this taxonomy is that it's very limited. It's very limited. First of all, taxonomies are dying. I have to say this. I would say in almost all countries, taxonomies are dying. Botanists in Malaysia, especially, I can count how many people that can ID the plants in the field. Zoologists, we are segregated into a specific group of organisms that we are unable to identify other group of organisms. Zoologists, taxonomies are dying. So, but with these tools, anyone can actually get access to understanding the taxonomy and also systematics classifications of this particular organism. So I've already talked a bit about taxonomy, systematic, and classification, but overall, this is the gist. All right, we are trying to understand where does a specific group of organism or animals or plants, in which group they belong to, because we cannot limit the understanding to taxonomy alone. In animal, we use International Code of Zoological Nomenclature to understand the correct taxonomical approach that we have that we can use to name or to change taxonomy of certain species. I believe in plant also you have the code. So that's only one subset of classifications, but molecular systematics and also systematic itself, we are trying to understand the relationships of everything that we are trying to study. So I don't think this is something that is very foreign in India, because I still remember throughout my journey as students, postgraduates, there is one scientist that I really look up to, right? Because I'm a primatologist, I study primates. So I'm not sure whether he's still there or not. I think he's an associate professor, Dr. Praveen Karan. I think last time, I think the affiliation is in Indian Institute of Science, Bangalore. So in 2008, Dr. Karan, he actually published a manuscript in molecular phylogenetics and evolution. In 2008, MPE was one of the best journal in genetics. So he published that. He was describing the taxonomy, the biogeography of Hanuman Langer, the leaf monkey that you have in India. So he's talking about the delineation of genus level, the biogeography, the disjunction, I would say in 2008, for an Asian scientist to do that, I'm very amazed with the achievement and published in a high impact journals. But if we want to look back, we just want to see the gaps. If we look back at one of the earliest study of molecular systematics in Asia, it goes back to 1990s. For example, Lisa Rosenblum in 1997, she actually discussed about the molecular systematics of the leaf monkey in Indonesia. So we can see, I can generally see the pattern of a 10-year gap between Asian countries and also the first world country, of course, American and also European, in terms of using these advanced tools to understand our wildlife. In 1999, the Americans and Europeans are already talking about mitogenomics, right? Mitogenomics in 1999. In 2000, the Asian countries are still learning how to use PCR to understand about biodiversity. So this is something that we have to look at and to reflect on, right? So going back to our molecular approach, right? I would like to talk about molecular phylogenetics, or we can say molecular systematics. So in terms of molecular systematics, we are trying to use the molecular tools, especially the DNA from the organism, for us to understand the relationship of the organism under study, in which we can look at population level, depending on locus or the markers that we use. We can study at the population, subspecies, species level. We can even look at genus and higher taxa rank, in which from this approach also, we can, from this data set that we generated, the DNA sequences, we can actually study the genetic structure of the population. We can look at the inbreeding coefficient, population subdivision. We can look at many aspects of population genetics, the minimum, for example, minimum spanning network. We can look at the haplotype relationship between one organism and the other. So this is something that is very interesting. But again, I would like to repeat, this is not new. This is 20 years of technology that are already, not maybe 30 years, that are already practiced by the first world countries. But we are still using it in Asian countries to understand our biodiversity, our wildlife, animals, and plants. In terms of phylogeography, of course, having the DNA sequences, or maybe the set, the molecular data itself, it allows us to use many bioinformatics tools. For example, we can generate a molecular clock trees, in which by using a certain recalibration point, a calibration point in time, using fossil data or geological data, we can recalibrate the evolutionary history, the divergence dates of a certain group of organisms. And then we can use this to understand the radiations and also the biogeographical events that are actually influencing this particular organism to what we have today, right? So one of the, I think, interesting thing to look at is also the advancement of molecular markers. So moving from a small data set in 1990s, 1980s, we're talking about RFLPs and all that, we moved to a single locus markers, right? I think the one of the most widely used are cytochrome oxidase 1, which is the barcoding region. And then that next come the multiplex PCR, where we can look at multiple locus at the same time. And then we have a nested PCR and long amplification method that allow us to study more and more of the DNA sequences of organism. But the advancement of markers is not only about getting more and more information or longer and longer sequences. It also the ability to use, I would say, much more different locus in understanding biodiversity, because for example, with the achievement and advancement in especially mitogenomic sequencing, in which we are sequencing the whole genome, we are able to generate and design a primer more specifically to different locus and different regions that can be used to understand much more details about our biodiversity. So talking about mitochondrial DNA, it's one of the most used markers in studying wildlife. We have cytochrome B, we have cytochrome oxidase C subunit 1, or CO1, we have D-loop or hypervariable region. So the diversity of locus that we can use for mitochondrial DNA is very big. And it's the same with chloroplast DNA, all right? In terms of barcoding, you have METK, RBCL, tRNL. Of course, even up until 2020, scientists are still debating which locus best used for barcoding. Same goes for mitochondrial, same goes for chloroplast DNA. And I think it depends on the expansion of database, because one of the big problems that we have in Asian countries is the database of these particular molecular markers. Because without a complete database, different scientists will come to different conclusion of which markers is the best. And this happened to my field of study, of course. But I would say that at population level, I prefer to use hypervariable region or D-loop region. And good thing about mitochondrial and also chloroplast DNA is because they are both what we call as DNA containing organelle that are present in the cytoplasm of gamete, in which it means that these two organelles, they most of them actually, they don't undergo recombinations, and they can actually serve as a pure information to divulge the lineage, evolutionary history of organisms as compared to nuclear DNA. In terms of nuclear DNA, of course, we know they undergoes meiosis and they have crossing over. And but not saying that the nuclear DNA is not useful, but it can give us a different perspective of evolutionary history. And it can, for example, to study the hybridization of organism, the hybrids, we may be looking at different locus in nuclear DNA, or maybe we want to look at the paternal inheritance. We want to look at the male evolutionary lineage. We can also use the Y chromosome and so on. So using these markers, all right, I know I have not got to my point yet in terms of advancement, but using these markers, we are able to exploit these markers using what we call as PCR. PCR is not something new. It was invented in 1983. The first world country scientists have been using it ever since. But like I said, it arrived much later, it exploited much later in Asian region. And I think one of the limitations, I think, this is my opinion, I think one of the limitations is because when this technology have to be exploited by wildlife researcher, because the integrations of different scientists, especially from biotechnological field and bioscience, are not well integrated in terms of wildlife and also conservation research. There's a big opportunity in this particular field because the gaps is so huge. The gaps is, I would say, I don't know about maybe plants, because I'm a primatologist. I study primates. So in terms of animal itself, the gap is very huge. We have so much group or taxa of animals, especially in Malaysia, that require much more examinations in terms of molecular approach. Just for taxonomy, population genetic is a different issue, biogeography is a different issue. So PCR, going back to this PCR, because I think this is related to my next presentation. So PCR is a method that was developed in 1980s. Because of the method, the author won the Nobel Prize in 1994, 1993. And the method is very simple. We want to amplify the DNA. So what we have, all right, so basically we have a starting material, which is the DNA, which is the first component of PCR. And then we need a primer, which is the second component. A primer is a sequence that are specific to a certain locus that are complementary to the DNA itself. And then we need DNA polymerase. In PCR, we use Taq polymerase, those that are harvested from Thermus aquaticus that can withstand high temperature. And then we must have our building blocks of the DNA, the nucleotides, right? The ATP, the A, T, C, and Gs, right? And with the right chemical environment, with the buffer and with the right temperature, PCR can actually proceed in 3 different stages. One is the denaturation, which is a stage where we are trying to separate the double-stranded DNA to become a single-stranded DNA. And after that is the annealing stage, which is the stage where primer that we actually mix together with the PCR cocktail will anneal to the designated DNA sequence. But we have to understand that, like I said, with the advancement of markers, this particular primer are also, getting much more advanced. Either it is too specific to a certain organism, or it might be, it can be more general or more universal that can target a bigger group of organisms. So after this primer has successfully annealed to the template, then extension process will occur and copy the DNA. So you start with 1 copy of DNA, you can end up with 2. So the concept is that the input DNA that we have in the starting DNA template is much more higher than 1, of course. And after 30 to 50 cycles, depending on directions, depending on the kit and chemicals that you use, we can end up having millions and millions of copies of DNA. So this is something that is very beneficial in terms of understanding molecular field of wildlife, right? Especially for those that are endangered or critically endangered. So after having that sequences, of course, we can move to bioinformatic analysis, in which we can have a sequence analysis to look at the parsimony informative characters, to look at the genetic distance and so on by using different algorithm. And then we can also have what we call as population genetic analysis. We can have minimum spanning network, like I said just now earlier. We can have so much approach in this particular analysis after we have the data set. And then we can have what, for example, here is the molecular clock, or the phylogenetic tree that actually have the divergence date at the nodes of each particular branch, and we can actually identify the evolutionary history of each subgroup as compared to one another. We can actually deduce the biogeographical theory using geological evidence and previous study about how a certain species arrive in a particular locations. So that is what I would like to call the first generation PCR. Okay, so in which even though the technology are old, but still up until today, it is very significant in terms of wildlife research, very significant, all right? It is able to amplify, like I said, millions to billions copies of specific piece of DNA with few starting copies. Why is this so important? Because this is very beneficial, especially for non-invasive sampling. Non-invasive sampling means that a sample that you can collect without actually harming or without actually interacting directly with the animal itself. So it can be feces mainly, it can be a follicle, it can be anything that is non-invasive. So with these non-invasive samples, usually the starting DNA is very small or very little. So with PCR, we are able to amplify this and we are able to get informations from these particular samples. And of course, lastly, the PCR is able to amplify old and or degraded DNA. Why this is important in terms of Asian countries? Because we have a lot of specimens. We have a lot of specimens, maybe not deposited in our country, maybe deposited outside, but we have these informations that is stored in the specimen. And of course, the approach of ancient DNA technique, the DNA extraction is quite different from a normal tissue or normal sample, but it allows us to study the DNA in this old specimen for us to understand more about maybe organisms or individuals that have already gone extinct from our ecosystem or from our country. Okay, so of course we have to also understand the expansion of database, right? I mean, looking 10 years ago, 10, 15 years ago, I think the database has expanded to a level that I cannot imagine. We have so much markers and locus and sequences that have been uploaded from time to time. Even a certain, I would say, high impact journal are now requiring the authors to upload the sequence of any phylogenetic studies to the general database before acceptance or even before being considered to review. So that's why I think contributed to the expansion of database. And with this, we have the ability to design primers, we have the ability to identify our samples and sequences and so on. So largely, I can divide this, which I say the applications of first generation PCR based wildlife research. So understanding this is quite, I would say, interesting because we want to look at what will happen next, which is the trend in the wildlife research right now. So the first one, of course, is classification, because like I said, the taxonomy in terms of wildlife or maybe plants itself is very limited in Asian and also developing countries. So having this method allow us to classify and also genetically identify the organism that we have in hand and correctly put them in the right group, which in case, because this is quite important in conservation and management, especially in wildlife, because we don't want to disturb the gene pool of the organism. And we have few cases already happening in Malaysia, especially involving translocation of pests and so on. And of course, we want to understand more about the organism using this particular molecular approach so that we can identify the boundaries, all right? Because the level of genetic diversity should not stop only at species level. There is also a concept of evolutionary significant unit, or maybe we can look at the differences in terms of unique gene pool of population from one population to another. And also, we are able to infer evolutionary history and phylogeography of the organism in hand for us to understand more about the organism. We are able to identify hybridized species. I think last year, our team published a paper on a slow loris, right? I think everyone knows slow loris. It's a primate species that are moving very slowly. So slow loris, Nycticebus kukang. So we believe previously that Nycticebus kukang is the only species that we have in the country, with addition of Nycticebus kayan, Nycticebus menagensis in Borneo. But in Peninsula, we believe we only have one Nycticebus kukang. But through molecular phylogenetic analysis, we are able to identify one subspecies that are present in the northern part of Peninsular Malaysia, which is Nycticebus kukang bengalensis. So this was never recorded before. And this is why molecular approach is important for us to know the boundaries. Or else, for example, let's say the management or the translocation program will simply translocate between one area to another without considering the differences in terms of their gene pool itself. And then we also managed to find one of the extinct subspecies, Nycticebus kukang insularis, in one of the islands, because we believe the subspecies are already extinct, but we found them again in one of the islands in Peninsular Malaysia. And right now we have 3 subspecies in Peninsular Malaysia. Previously, it's just one. Over the last 3 years, with this particular exploitation of molecular tools, now we have 3. So we know that we have to manage them well, because the population for bengalensis and also insularis are very small, and we have to pay more attention to these species. All right. Same goes to management practice. This is evident in the case of, in our country. I don't know about India. I would like to know later. In our country, Asian elephants is one of the center for human-wildlife conflicts. All right. So they are always reported in oil palm plantations. They are always reported sometimes entering villages, crossing the highways and so on. So the authorities, since the last 15 to 20 years, have been translocating them all over the countries, right? All over Peninsular Malaysia. So what we have, what we can understand from molecular approach, this was a study made by our team in which we know now that in Peninsular Malaysia, we have no unique gene pool of Asian elephants, in which we cannot pinpoint these particular populations are from which state or which population, because everything is mixed up, right? Because over the last course of 20 years, this translocation program has been going on and on, in which they simply capture elephant from one state and translocate them to another state, and they have been moving them around the state, around Peninsular Malaysia for over the years. So now we don't have any unique gene pool for these Asian elephants. So this is another thing that we have to look at using molecular tools. And then also long-tail macaques, all right? I believe long-tail macaque is also one of the issue in India, all right? So long-tail macaque also is the same thing, because translocation problem, in which right now we don't have any unique gene pool of the long-tail macaque in Malaysia, with exception of few islands, all right? So other application of first generation PCR is also wildlife forensic. This is something that is very interesting. This is a picture from my postgraduate student that visited Aboriginal village in Malaysia, because the idea is to get information. Number one is to get the idea of distribution of animal is quite challenging in Malaysia, due to funding, due to manpower, and due to the large area that we have in certain states. So what we do is we go to this Aboriginal village, all right, in which they still practice their culture, their traditional knowledge, and we actually interview them, all right, and we collect specimen, all right, either skull, either tissue, either whatever skins that they have preserved over the years, whatever bones that they have, and we bring it back to the lab, and we use DNA barcoding technique, and we identify which species do they belong to. And we found out that it's quite worrying that the Aborigines are consuming a lot of endangered and critically endangered wildlife. So this has never been reported before. This is what we call as ethnozoology, molecular ethnozoology. So ethnozoology simply before this is just, you go to any villages and just interview them to get information. But now with the addition of molecular tools, we are able to pinpoint the pattern of wildlife consumption among these Aborigine that we have in Malaysia. All right, so this is the application of first generation PCR, but all of this would change with the arrival of next generation sequencing. So the arrival of next generation sequencing is not new to the world. It has been around since 2000, but I would say the access to this technology in Malaysia in wildlife research, don't get me wrong, in wildlife research, is quite worrying. Of course, we have few teams and institute working on NGS, next generation sequencing in Malaysia, but they are more focusing on human perspective, diseases, maybe agriculture, but not on conservation. So basically, if I just want to give a brief overview of next generation sequencing, it is a technique that that allow us to get much more data from the samples that we have. We call that as high throughput sequencing. It uses different modern sequencing technologies because we have different platforms, like you said, like you can see here, Illumina, Ion Torrent, Applied Biosystem, and so on. But the idea is the same, is that to have a large data set, DNA sequences from the samples. And it's not limited anymore to what we call as amplicon sequencing. So previously, in the first generation PCR, you have to amplify the DNA, and then only using the amplified part, or we call as amplicon, then only we are able to sequence them using Sanger sequencing. But using NGS, you can have two methods. One is you can have what we call as direct sequencing after you have conducted your library preparation. And then you can also have amplicon sequencing. But using amplicon sequencing in NGS, it's not limited anymore to a single locus or single species. So the data set is very huge. So biodiversity survey are also possible due to this technology for limited cost and minimal efforts. And this, I will talk more about this in my environmental DNA section. So due to this advancement that we are able to sequence more, for example, a mini-seq system by Illumina are able to sequence up until 7 gigabase of that is unthinkable. I don't know, to me that is unthinkable to I mean myself for the last 10 years, because we are dealing with I mean last 10 years, if I'm able to sequence 1400 base pair of the DNA from CO1, for example, of primates, I'll be very happy. The chromatogram is good, there's no noise, I'm able to align all sequences and come up with my phylogenetic tree, I'm happy. But now, I'm able to generate in my lab 7 gigabase of DNA sequence from the samples. And this is made also possible not only by the expansion of chemistry that are available in the technique, the expansion of kit that we have in the market as well, and then it is also made possible by the affordable and lower cost of sequencing, I would say, to everyone available. And And I would say because of the expansion of bioinformatic tools available. So we have many tools available. If it is a user interface based, you have CLC Genomics by Kaizen. We have NextGene, Genius. If we are looking at maybe pipeline based, we have a lot of pipeline available, free pipeline available on the internet. Chime. We have so many other bioinformatics tools that can be used to understand our NGS data. So I would roughly divide the NGS in terms of conservation. I know NGS has so many other applications, especially in health and diseases and so on. But I would like to just talk about conservation of organism in terms of NGS, right? So the first of all is that it allows us to expand mitochondrial DNA as marker. Before this, like I said, in the first generation PCR, we have to amplify a certain locus that we want, a certain region of interest, certain markers, and then we will have an amplicon. This amplicon will be sequenced. But now using NGS, we are able to sequence the whole mitochondrial genome, the whole, in mammals, 16.5 kilobase. The whole genome, we are able to sequence them using this technique. And we can come up with what we call as mitogenomic phylogeny. It's no longer what we say an argument in which one scientist use CO1, the other one use D-loop, the next one use cytochrome B, and they are fighting about their outcome. This is because they are using different locus. But by using mitochondrial genome as a whole, we are able to generate, I would say, a very large data set for us to understand the relationship and evolutionary history of organisms. And then we can also use shotgun sequencing, all right, in which we are breaking down, especially when talking about mitogenomic, we are also using shotgun, but I specifically put this in a different section because this is something that is very, very useful in using non-invasive sample. If we just want to have a very high throughput mitogenomic data, using tissue samples are very useful. But using a deep sequencing shotgun method, we can use non-invasive samples such as fecal sample that allow us to have these small fragments of mitochondrial DNA that are sequenced throughout the samples, regardless of the organisms or the host DNA, maybe microbial DNA and so on, and we are able to reconstruct these particular fragments using bioinformatics tool. And of course, this is something that I really hope in the field of conservation, we will expand into this, which is a meta barcoding, right? So like I said previously in the first generation PCR, if I collect a sample from the Aboriginal village, I use the tissue and I use DNA barcoding technique, I can pinpoint what species that the skin or maybe bones belong to. But meta barcoding, you are barcoding everything that you have in your samples. For example, the currently exploding, the last 5 years have seen the explosion of microbial gut microbiome or microbial community sequencing of many samples that use 16S rRNA to identify the microbiome that you have in your samples. I'm also interested to see if the diets of organisms can be analyzed, which by using meta-barcoding of chloroplast DNA using METK, tRNL, or maybe RBCL to understand what the herbivore are actually feeding on in the wild. And also, it's very important also for us to understand about zoonosis perspective. Of course, it's not my field of expertise, zoonosis, but I think this is something that should be explored by all of us in Asian countries that have a rich diversity of organism, wildlife, plants, and so on. And we are always exposed to the emerging threat of this disease from wild animal. So this is another thing that I would like to share, right? Environmental DNA, something that is very, I would say, not many studies are being conducted in eDNA. I don't know about last 5 years, but I would say the methods are not really well developed right now for all of us to use. The primers are still changing from time to time, especially for animal barcoding, meta barcoding. But it's a good idea for us to use these particular tools of environmental DNA to have this large scale monitoring of biodiversity. Now imagine, previously, if I want to know what, for example, the fish or a freshwater fish that I have in certain lakes or rivers, I have to catch them. I have to put up traps and maybe nets and I have to catch them physically. But using environmental DNA, we believe that organisms are actually secreting all these DNA traces in their environment. For example, their tissue, their blood, their feces in the environment. And all these DNA are suspended in the environmental sample. Mostly used are soil and water. But let's talk about water, because water is the most used environmental DNA samples right now. So by collecting the waters, filtrating the waters using cellulose paper, nitrate cellulose paper, and using the particular filter paper for your DNA extraction, you can have all the DNA that are present in the water samples. So we have few projects in Malaysia exploring this. One of my postgraduates are also exploring this, the viability of doing this. Because if you are trying to map the DNA that you have in your ecosystem to the database, it comes back to the whether or not your database is comprehensive. That is one of the limitations that I see about these particular tools in Asian countries, because we don't have a complete database. For example, talking about plants, I'm trying to understand about plants, or maybe I have samples that have a lot of different sample of plant in that particular samples, and I'm trying to reference them to my database. The problem is, because I've tried meta barcoding before for certain species of herbivore, all the species that come back to me are from Madagascar, from Africa. It's not from Malaysia, because the database for plants in Malaysia are not well developed. Same goes for CO1, or maybe for vertebrate or invertebrate barcoding, because the database is not that well developed. So we are unable to pinpoint accurately that this sequence belongs to which particular organism. So in the detection of environmental DNA, I don't want to limit the discussion to NGS, because I have to acknowledge PCR, qPCR, and multiplex PCR are also used to understand the detection of environmental DNA, in which if you're using PCR, we are using specific amplification. Recall back my previous slide about specific primer. So we are using specific amplifications in which we can have large copies, but it is extremely sensitive to contamination, and it can only be used for single species approach. For example, it is being, I think, announced in the House of Australia in the parliament where they announced this particular method to detect and monitor their carps in their ecosystem. So that is a single species approach. But using qPCR, we can actually not just detect, but we can actually quantify by using the software and also calculations that you have in your qPCR system. So you can actually quantify the DNA that you have. Multiplex PCR, on the other hand, allow you to identify multiple targets in the single PCR reactions, but NGS would be the best. The next generation sequencing would be the best because it sequences everything that you have in your samples, and it allow you to use your bioinformatic tools. In my lab, we use CLC Genomics. So, but the database, we have to construct them. We have to customly develop the database in order for us to have the reference point. But NGS would be the best because you are looking at the sequence level, not only detection, not only quantification, but you are looking especially at the sequence level. You are trying to barcode them to the database, and you are able also to quantify all this data for your samples. So this is I would say, the method that I don't know whether this would be the most comprehensive, but we developed we actually designed this particular figure for us to, and because we always explain to stakeholders, we always engage with stakeholders and authorities and going and promoting all this technology for them to get involved with. So we would start with sample extraction. All right, let's say you have sample from water or soil, and then actually you can have what we call a shotgun sequencing here without amplicon or amplification using PCR. You can always start using shotgun sequencing. But the problem with that is that you need a lot of money to do that because you need a very deep sequencing. You need, because you are sequencing everything that you have without including those region or markers that you won't be using for your identification. So that's wasting a lot of resource, right? But if we are using amplification or PCR technique, and let's say, for example, not just NGS, this is just a figure on environmental DNA. So let's say you don't want to sequence, you don't want the data. So if you don't have to sequence the data, do you have to quantify them? Do you want to know how much it is present? So you can use qPCR to quantify this. If you just want to detect whether or not they are present in your samples, you can just use a normal electrophoresis technique to see the amplification, whether it happens or not. But this is the interesting part. If you want to sequence them and you are dealing with multiple species, for example, you are using primer to understand the freshwater fish that you have in your samples. So you will use what we call as meta barcoding. You will sequence them using NGS technique, high throughput sequencing, and then you will be conducting your species identification by referencing your data set to your database that you constructed, okay? So in terms of review that we have over the last, I would say 20 years that we have reviewed the data, we can see that fish, freshwater fish, and also invertebrate are the largest group of organisms that are being focused on in terms of environmental DNA. And you can, in terms of locus, right, you can see here 18S, this one is 12S, this one is CO1. CO1 is much more used in invertebrate. This is due to the fact that each organism, or maybe different group of species, they have a different level of database in the system. So thus, for example, barcoding invertebrates, right? Mammals, for example, we rely more on CO1. Same goes for a certain species of invertebrate. But in insect, we use 18S, 16S. In fish, for example, 12S. So it depends on the organism that we want to study, and it does not actually tie us to any specific locus that we have to use for all environmental DNA. So this is an overview of the wildlife that we have in terms of their locus that are being studied using environmental DNA technique. But in terms of environmental DNA assays that are being used throughout, I don't know why I have the, maybe someone is drawing on my slide, but okay. So in terms of the assay, all right. In terms of the assay, we can say that majority of the assay use what we call as next generation sequencing Illumina platform, in which it is a sequencing by synthesis chemistry. It also offer a lot of options on the library prep and also amplicon sequencing kit available on the market, but also other techniques are also used, especially the qPCR or the real-time PCR. In terms of platform use, like I said, Illumina mostly used as compared to other platform due to the fact that they have a variety of different machines and instruments that they offer to the market. And also, when we talk about platform, we have to understand that it's not about one platform is better than the other, but you have to understand that each platform or each technique, they have a different ability to conduct single end sequencing, paired end sequencing. They have a different error rate. For example, in Illumina, they use 5x as their reference for their quality control. So they have a lot of things that you have to consider in terms of sequencing. So I would say in terms of future directions, we need a lot of increasing expertise in terms of next generation sequencing in wildlife. Also other conservation of other organisms. And also we need more study on microbial and zoonosis interactions. We have to look at ancient DNA samples for us to understand more about our specimen, especially, and also conservation genetics, management and implementation in the country, and as well as integrating these tools for future study for wildlife and also maybe for other organisms in the name of conservation of biodiversity. I think that's all for me. Thank you very much.
Thank you, sir.
Welcome.
Oh, hello.
Thank you, sir. It was a wonderful session.
Yes, madam. Madam,